Hi, and welcome to this lecture on environmental adaptation of animals. This is the last phrase of animals and environment subject, and it's called on adaptation itself. So now we know that, okay, what is an animal and what is the environment, and now we will deal with the adaptation. So what's the adaptation? Well, when we have a population, there's a genetic material that is more or less popular. So these alleles, so different versions of genes can be more or less popular. And when it is isolated, these mutations can change the normal code in the population. And these mutations, they can be adaptive, or their frequency can change also by chance. And if they are adaptive, some allele, they should have high frequency in the population because they are giving some benefits for the selection. But, however, quite often we have seen some non-adaptive evolution in the animals. So all the differences, whether it's just that the animal looks different or the genetic code is different, it's not adaptation. Well, I give you now an example. Some time ago, I was comparing genetic difference in one ion channel protein and compared different organisms. They all are vertebrates, and thinking, okay, that over there is a part of the protein segments of some common terrestrial animals, and then on the right hand side there are two protein cloned from rainbow trout and one from Crucian carp, and then comparing one amino acid by one amino acid, that okay, what are the differences between them. And in the center you can see that on the yellow there is cysteine. That is a very particular amino acid because it can interact with other cysteines. And it's can be found in all the terrestrial animals, but not in these aquatic animals. And of course, I was very excited that, okay, this is the magic bullet that is making the difference between fish and terrestrial animals. But unfortunately, it was not very important for the functionality of this ion channel. So it was just a chance that, okay, because it's so long time in the evolution, so it's just have been changed during the evolution. So it's kind of non-adaptive evolution. And so, and non-adaptive, in 1970s, still many physiologists referred all the traits. So this animal looks like this, so it must be adaptation, or it has this and this kind of organs, that's adaptation. Or when we were comparing genetic code or whatever, okay, these have more this protein or this hormone or whatever, always saying that, okay, this is adaptation. And it was not tested. But then in late 70s, they were finding out that, okay, actually natural selection is only one part of the driving force of changing these frequencies in the genetic code. So there can be genetic drift, especially in a small population, because so the chance can have the major role in causing the differences in gene frequencies. For example, if we have a population where we have two different alleles, A and B, and then by chance half of the population will die. So there is always a chance that the one allele will disappear completely, especially when the population is small. And also, when we have a small population, most probably there have been only few individuals that have ever entered in that environment to start the population. And they might not have the optimal genetic code for that environment. And this is something that we have seen a lot in human populations. Like in Finland, we have only 5 million people. So when this humans have not moved around, they are making some kind of small populations and local populations and causing certain diseases that are typical in Finnish population rather than some others. And of course, because if they are diseases, we can understand that, okay, this is not adaptation at all. So we can have different allele frequencies common, although they are not adaptate at all. And there are some reasons for that. First of all, the trait can be correlated with another trait that is favored with the natural selection. Previously, we thought that, okay, the number of genes determines directly the number of proteins, and one protein is regulating only one trait. But nowadays we know that, okay, these genes can be making several different proteins, and these proteins can be related on several different traits or properties of this animal. And on this, there is a good example of one mosquito which had one allele that caused it to survive against one pesticide. And okay, when people were using this pesticide, this allele was getting more popular, the frequency was increasing, and this same allele for some reason was giving this mosquito also tolerance for cold climate. So it was spreading. Or then the other example on this non-adaptive evolution is that this trait can be evolved as an adaptation to an ancient environment. And for example, fish, whether they are marine fish or fish in lakes, they have quite dilute blood in their veins. And in marine environment, this is not very good benefit, because some other animals that are living in the oceans, they have much more salt inside their body, so they don't have so much use energy to pump out all the salt from the gills. But these marine fish, they must do it, but because it was planned for something else. Okay, how we can know then that something is adaptive? Well, we must test it. The old school method was just to direct observation and natural experiment. So just watching your backyard and seeing that, okay, what's happening there. And this is not very common nowadays. But it's still, in the good old days, the way that it was found that, okay, industrial melanism, that, okay, some butterflies in UK were changing the color because there was so much pollution caused by these factories. So when they were changing the color, they were hiding from the predators, and then this allele was getting more popular. Of course, uh, nowadays we do a little bit differently. Because we can't make the direct observation, we can also always make comparative method. We can compare related and unrelated species in similar and dissimilar environments. So do all the animals that are living in this region have this and this kind of properties? And are these properties different in cold climate whereas in warm climate? Then one interesting way is just, okay, put the animals in the laboratory for several generations and expose them for some environmental factors. I have read some articles where they have had some fruit flies and exposed them in cold climate to see that, okay, what's happening there in several generations. And it works well in fast breeding animals, but because of the business reasons, it can't be done in larger animals at all. Then we can also test in single generation to see that, okay, what kind of individual variation we have. And this is something that is done a lot in medicine to find the correlations. That, okay, some animals are better in sport, and also these people are better in they live longer than those that are not very active in the exercises. But then later on, it was found out that actually it was not the sport that was causing this, but they had a little bit different alleles in their genes. So this is something that is done in medicine a lot, but it's quite often difficult to find out that, okay, what is the function that is causing this. Okay, then something that is not done a lot in comparative physiology, but for example in development biology, this is the trend that, okay, we can create the variation. We can make a knockout, and with these gene scissors, we can make knockout on any animal on this planet easily, on any protein. So we can test that, okay, if we knock out, remove one protein from the individual, can they still survive, or will they die? To see that, okay, and this is a very simple way. Then it's quite definable that, okay, it's an adaptive mechanism or it's not. Then, of course, we can study the genetical structure of natural populations. So to see that are there any changes in the phenotype frequencies along the environmental gradient. And this is also something that is very new in the comparative physiology, because it takes a lot of time. We need a lot of samples, we need a lot of locations, etc. And then last but not least, we can also make phylogenetic reconstructions. So we can compare, for example, I should just show that, okay, there was these differences in the protein sequence. So we can make a family tree of the groups of relative species to see that, okay, when something has been happening, so, okay, fish have this kind of protein, and these terrestrial animals that have another kind of protein, or then we can see that, okay, are there any common ancestor at all? So might be that sometimes we have similar in that have in animals, but they've been developing independently. Okay, some examples. Comparative method. We can see that all terrestrial animals, they have independently evolved some kind of breathing organ that is invaginated into the body. So whether you are human or insect or land snail, you have some kind of lung or mechanism inside your body to get oxygen and carbon dioxide moved. And that's why this comparative method is very simple to make. It's just the idea is that okay, many kind of these animals alive today, they provide examples of outcomes of evolution. Then the other way is to check the individual variation. For example, in these mice, so you can measure the maximum rate of oxygen consumption and see that okay, the average is there on 19 to 21 milliliter of oxygen per gram per hour. But then still some are better. They are able to use 24 milliliter, and some are consuming only 16. And this difference means that actually these that are using a lot, they are also producing a lot of ATP, and perhaps they can run faster. So there can be, in the population, can be differences between the individuals. And then we can make a lot of experiments in the field to see that what kind of traits are changing along the environmental gradient, because animals can have several potential traits that could be benefit in, for example, cold climate. But if they are not changing along the environmental gradient, it's not adaptation. So we must have several locations to see that which factors are changing and which has the high genetic diversity. For example, over here we have in US made with the house mouse to see different the genetic diversity in different factors. So what is the body temperature? What about the amount of brown fat? What is the amount of what is the body size or nest size? And as you can see that there are latitudinal trends of increasing size of body and the nest from the south to north in these traits. But on these other ones, although we in common sense, it makes common sense that okay, the brown fat is helping you in cold climate, but it's not changing. So it has low genetic diversity. Thank you.